So in the last class we have talked about receptors. So we have seen that there are various types of receptors. Some receptors externally bound, we call them the exterior receptors. I'll just go back and revise. Some receptors detecting changes in the internal aspect of the body, we call them interior receptors, and the other receptors which detect position of the body. Now among these we have general and special. So classification for general senses like touch, pain, pressure, temperature, all of this. Special senses include vision, right? There's sound or the auditory transduction, that smell, olfaction, right? All of this. Balance is also a special sense. So we have seen all of this. We have also classified receptors on the basis of stimulus energy and on the basis of adaptation. Right? So structures also we have seen. These are the various types of tactile receptors, the receptors which are present on the skin. And you can see clearly there are two receptors which start with the word M, Merkel disc and Meissner's corpuscle. Both of them are superficial receptors. They are placed in the superficial aspect of the skin. And the other ones, they are the deeper ones, Ruffini's end organs and Pacinian Corpuscle. One more way to remember is about which one is slowly adapting and which one is fast adapting. So the corpuscles are rapidly adapting receptors. That means they become active when the stimulus is on and the stimulus is taken off. During that period when a constant strength of stimulus is maintained, their response is less, rapidly adapting. The other ones are slowly adapting receptors. So enlarged view of the Merkel cell and the Merkel disc. Now this receptor is actually present in clusters, right, on the center of the papillary ridges. So you can all see the papillary ridges on your skin. So these receptors are located as clusters in the center of the papillary ridges. So these are called as Dome Receptors. This name also you might come across in your books. So these are clusters of Merkel disc. And these cells have the smallest receptive field. So that is why their ability to localize and to discriminate between two objects or two stimuli given very close is very great degree of discrimination is there. So this property is also called as two-point discrimination. Okay. We have also seen an enlarged view of the Pacinian corpuscle where we also highlighted that the center of the corpuscle is where the nerve ending is present, right? And the first node of Ranvier is within the corpuscle, second node is outside the corpuscle. Signal transduction starts within the first node itself. We will come to this in a bit. We have seen epicratic and protopathic receptors also. Okay. So now about the functions. This also we have seen. Meissner's corpuscle, right? The first one, they detect touch and vibration. So why did I mention texture over here? So Meissner's corpuscle, they are present on the edges of the papillary ridges of the skin. So location wise you see the slight difference is there. So they are present on the edges of the papillary ridges of the skin. So that is how whether a surface is rough or a surface is smooth. So this is the receptor which identifies this texture. It is a variety of or a form of touch, but we can still attribute a special touch. So this receptor is concerned with texture. Okay. Deeper ones, Pacinian corpuscle, deep pressure and vibration. Ruffini's end organs also concerned with pressure, right? And Merkel receptor is concerned with sustained touch. So one very interesting thing you can also observe here. So yesterday we have talked about this, that touch, pressure and vibration, these are various forms on the same spectrum. So when a tactile receptor is stimulated, right, superficial ones, if they are activated, so the body perceives them as touch, right. So if the pressure, so suppose the stimulus is strong enough that it also deforms the receptors present in the deeper aspects, same stimulus, you can hold like this very lightly, so body identifies this as touch. right? So if you hold us at the same place with a greater amount of force, so now the body identifies this as pressure. So क्या difference था दोनों में? In the first instance, the mechanical force was slight, so only the ones in the superficial aspects of the skin were activated or they were stimulated, so they detected sensation. So this was then identified as touch. When a greater degree of mechanical force is given on the skin, stimulus is stronger, so much so that it is able to deform these structures. In the deeper layers, the body perceives this as pressure, right? And if it is a cyclical stimulus, so then the body will identify this as vibration, right? And you can see that vibration can be detected both at the superficial aspects as well as the deeper aspects. So as I said, an easy way to remember is the ones starting with M are superficial, and the ones having corpuscle, they are the ones which are rapidly adapting. So vibration mein what is happening? So vibration is a cyclical stimulus. एक point पे there is a stimulus, then goes off, again there is a stimulus, again it goes off. So for a vibration to be identified, it is essential that the receptor should be rapidly adapting. On off should be rapidly be identified as separate. Okay. Pain के जो receptors है, I have said yesterday also, these are bare nerve endings, free nerve endings without any form of structure associated with it. Motion can be detected from the hair follicle receptors. Right? So movement, any form of movement on the skin. Now let us start with the process of transduction. So transduction is the basic process by which all forms of stimuli are converted to some electrical signals. So there are two parts of this process. The first part is the generation of a formation of a generator potential or receptor potential, and the second step is how the receptor potential gets converted to an action potential. So why do we need this? So both of these are important, right? This question might come to your mind that why not in the first instance एक ही बार में action potential क्यों नहीं बना, right? You would have solved immediately, it could travel on long distances and reach the brain, because long distance communication के लिए we need action potentials. Why the formation of a receptor potential to begin with? Why not an action potential at the outset? So there are reasons for that. Now all of this, as I said yesterday also, were identified by studies on Pacinian corpuscle. So placing two electrodes, one on the surface and on the center, and recording the electrical changes in the nodes of the Ranvier. So this was how everything was identified. So initial response, as I said, is a receptor potential. Receptor potential, you should remember, is a form of local potential, and there are very salient differences between a receptor potential and a local potential. Okay, why did I use these two terms, generator and receptor potential? They are the same thing, local potential itself, which is happening at the receptor. Now if the receptor is a nerve ending itself, we call this generator potential. If the receptor is a special type of epithelial cell synaptically linked with a sensory neuron, that initial change we call as receptor potential. But both of these are happening at the receptors only. Nomenclatures are decided. What is the receptor? Is the receptor a nerve ending itself or a epithelial cell linked with a nerve ending? So that is the reason there is a difference of the nomenclature. But the bottom line is, this is the initial response when there is a stimulus. So whenever you give pressure, what happens? So let us think about it. So pressure or any form of, say, tactile sensation, touch also, right? A slight touch also can do the same thing. So it will deform some layers of the Pacinian corpuscle, and eventually this force or pressure will be transmitted to the neuron, right, or the nerve ending which is there. Now here what happens is, there is opening of stretch sensitive cation channels in the neuron, and cation channels, once they are opened, so what will happen? There will be influx of cations, sodium, in some cases calcium, majorly sodium. So what has happened is, in the neuron now you have a depolarization, right? So initial response is a local potential. Now what are the forms or properties of local potential. So local potential follows a graded response, right? These are concepts you have learned in the first unit, right? General physiology. So what is the meaning of a graded response? What does it mean? Gradation, when I say, what does it mean? The response will vary along with the strength of the stimulus. If you give a slight strength of the stimulus or a low intensity stimulus, the response will be small. If you give a stronger stimulus, the response will be stronger. So this type of response where gradation is possible, we call this as graded response. Graded response is a property of the local potential. Action potentials, they do not follow local this gradation. Why? Action potentials follow all or none law. So if you give a sub-threshold stimulus, you get no response. If you give a threshold stimulus, you get maximum response. Even if you keep on increasing the strength of the stimulus, response is already maximum. Response does not increase further. So clearly, there is no relationship between the strength of the stimulus and the response in case of action potential. Right? When I am talking of a daily life, it is important that we should know what is the intensity of the stimulus. Receptor level mein, this property is made possible because of the formation of receptor potential. Stronger the stimulus, higher is the magnitude of the receptor potential. So that is why the things are like this. Right? Okay. Now once the depolarizing change has started developing and say it reaches around 10 millivolt of depolarizing change. So I am talking of the generator or the receptor potential. So receptor potential, local potentials like this, they can spread to small distances. They cannot spread over long distance, but they can spread around, right? Try to remember about the property of decremental conduction. Initial आसपास का जो area है, that can be stimulated or they spread to a certain distance, but then the intensity drops exponentially, right? So the first node of Ranvier is able to be depolarized by this local potential. So because it is at a very close distance. So I am talking of the bare nerve ending here, which is present in the center, jahan pe receptor potential had developed. Now because it can spread to small distances, it reaches the first node of Ranvier over here, right? 10 millivolt ka agar charge or depolarization, if it has reached there, this will trigger the formation of an action potential in the first node, right? Why this 10 millivolt of cutoff, why this much? Because every tissue, every neuron, they have a firing level. So 10 millivolt is the firing level. So as soon as in the first node of Ranvier this much has reached, so it will trigger the formation of a action potentials. That is the firing level or the threshold level. Action potential by definition says that there is a depolarization followed by a repolarization. So initial जो receptor potential का magnitude था, if it is 10 millivolt only, so you get one depolarization followed by one repolarization. You get one action potential, one impulse in the nerve. Suppose it is stronger, 30 millivolt, 40 millivolt, 50 millivolt of change. So what will keep on happening? Receptor potential is large. So just after the repolarization has happened, immediately again in the node a second action potential is triggered. And that also is followed by repolarization. Again a third action potential is triggered. So larger the magnitude of the receptor potential, that many number of times the node will be triggered to its firing level, and that many number of times there will be action potentials or impulses, right? So is this part clear? So larger the magnitude of the receptor potential, more is the number of action potentials that can be triggered. So it is the first node of Ranvier which converts the graded response of the receptor to action potential in the nerve. So you see the beauty of this. Receptor mein response should be graded so that the body at its entry point can identify what was the strength of the stimulus. So graded hona hi chahiye, right? And that also needs to be converted to action potential so that the brain also knows it. Eventually should be converted to the conveyed to the brain. So what do we see here? The number of action potentials produced, or frequency of impulses produced in the afferent nerve is decided by the magnitude of the receptor potential, which in turn is decided by the strength of the stimulus. Right? So if I ask you, how does the brain know what was the strength of the stimulus applied, pain stimulus or touch stimulus applied? So number of action potentials produced or reaching the brain, number of impulses reaching the brain, so that tells the brain what was the intensity of the stimulus, right? So sensory coding. So sensory coding is the process where a stimulus as such is identified as a recognizable sensation. Recognizable sensation means we talk of all these different type of attributes, right? So what was the modality of the stimulus, location of the stimulus, where exactly in the body, or was it acting on outside the body? Stimulus acting from outside the body, we call this teleceptors. Do you remember some examples of that? I gave some examples yesterday. Teleceptors के example, stimulus बाहर है, not in contact with the body. What example comes to your mind? Light, right? Light is present outside, right? Is not coming in contact. But receptors are able to detect even when the stimulus is present at a distance. Okay. Intensity of the stimulus out, not. You are late. I don't know the reason. Please leave. Intensity and duration. So these are the 4 attributes of the stimulus that we identify, right? So I am giving again the example of this 4 tactile receptors. Location. Modality, the first attribute, we classified the receptors, remember. So classification of receptors on the basis of modality. Some receptors were mechanoreceptors, chemoreceptors, right? Then what else do you remember? Nociceptors, thermoceptors. So every receptor is specific for a certain type of stimulus, right? So this is called as adequate stimulus. So what adequate stimulus means, if that particular stimulus is given, the threshold of activation is very less. So they will respond adequately or optimally. But this also does not mean that it cannot be stimulated by other receptors. So what does, again, this is a very confusing thing. So let's take the example of photoreceptors, rods and cones which detect the amount of light in the environment. So adequate stimulus for a photoreceptor is light. But did it ever happen to you? If someone punches you, or you get a sudden blow in your eyes, right? And sometimes we feel like flashes of light. ये हुआ किसी के साथ या नहीं? Has it ever happened to you? It has happened to me. If you get a blow or any type of sudden mechanical force on the eye, right? There is a sudden flash of light. So that means your receptors they were also activated by some other type of stimulus. But difference क्या था उसमें? Threshold बहुत ज़्यादा था. So it had to be sudden and it had to be significant. So you touch like this, you will not feel anything. So this is a common example. One more example that comes to my mind is thermoreceptors. So let's take the example of receptors which detect cold temperature, right? So if the temperature, say let's say of the water you're holding, it's around 20 degrees Celsius, तो आपको लगेगा ये ठंडा है. If I keep on reducing the temperature of the water, say I take it down 5 degrees lesser, so you feel it is colder. 12 degrees Celsius, you feel it is more colder, right? But the moment it crosses 10 degrees Celsius, below that, now this becomes cold, and along with that there is pain, right? होता ये कि बहुत ठंडा जब है, you cannot hold ice for much long. Why? You start developing pain. So receptors जिनके लिए the adequate stimulus was temperature, now they are producing pain. So these are examples where concept of adequate stimulus comes in. Second aspect that you should know here is there is a law, Muller's doctrine of specific nerve energy. So this law states that when nerve from a particular sensory receptor is stimulated, anywhere along its course to the brain, the receptor से brain तक anywhere, if a particular nerve or a pathway is stimulated, right, sensation evoked is that for which the receptor is specialized, no matter how or where along the nerve fiber the activity was initiated, right. So say for example a nerve carrying, so connected from a Pacinian corpuscle, it is carrying the sensation of touch and pressure to the brain, right. It was here, suppose an example I am taking, somewhere from around the hand. There is some type of, let's say there is a tumorous growth in the elbow which is impinging on the nerve. So receptor पे one no activity is happening, nerve के pathway में there is some type of impingement which is happening. The sensation however is produced is a sensation of pressure or touch. The example of color vision. So there are three types of cones, this much I expect you know, blue, green and red, right. So this law states that if you stimulate a nerve coming from a green cone, so anywhere along its course, sensation produced will be green color. If you stimulate a cone, nerve fiber coming from a red cone, anywhere from the receptor to the brain, nerve में कहीं पर भी stimulation हो, sensation produced is of red color. So this is what this law states. So this is again also an important question from the exam point of view. It's asked. Next aspect, location. Where in the body do we find the stimulus? So to understand location again, you need to know about 2-3 things more. Sensory unit, receptive field and the punctate character of sensations. So what is a sensory unit? A sensory unit is a single sensory neuron or an axon and its peripheral branches which are connected to the receptors. So a single sensory unit can have more than one receptors. So single axon and all its branches form a sensory unit. The area of the body from which a single unit, a sensory unit collects its sensations or receives its sensation is called as a receptive field. So it is a spatial distribution. Spatial comes from the word space. Spatial distribution from which if you give a stimulus, a response is produced in a given sensory unit, right. What is the punctate character of sensation? Now if you map the entire body using a very fine strand of hair, millimeter by millimeter, you will find that at each point where you have a touch receptor, you appreciate the stimulus, right. But this is not a continuous thing. Adjacent to this, you will have some areas, intervening areas, where even if you put a stimulus, there is no response. So punctate means spot, small, right. So the receptors are present all over the skin, right, and intervening areas you have to understand that they are present as small spots all over the skin. Sensations can be produced only when the skin overlying the receptors are somehow activated or stimulated. Intervening areas, they do not have any type of sensation. So this is what this one means, right. And receptor fields, they overlap except for thermoceptors where the hot and cold spots all over the body, they are very distinct, right. So other sensations or modalities, there is a degree of overlap over all the receptive fields. So taking all of this into knowledge, now let's understand how the body knows the location. If you put a, so can you close your eyes again? Now with your right index finger, if you touch the center of your left palm, so can you appreciate the location of that? Stimulus कहाँ पे था? You can, right? Even without seeing, you can appreciate. If you stimulate, say some other area, say the flexor aspect of your elbow, even without location, without opening the eyes, you can appreciate the location. So that means even without seeing, we know where is the location of the stimulus. So brain somehow knows, right? So this is something called as sensory homunculus. So in the post-central gyrus of the brain, so this is the central sulcus, right? Anterior to this, you have the precentral gyrus, which is the motor cortex, and posterior to this is a part of the parietal lobe. You have a small strip in the cortex where the entire body is mapped, right? So you have the map of the whole body, topographic map of the entire body. This is called as homunculus. Homunculus means a little man, right? And you also see that, like, it does not look like actual human being, right? You see the face is disproportionately large, hands, the fingers are larger, but if you see the limbs are small. Why is it so? Why do you think it is like that? So greater the tactile sensitivity or acuity, जहाँ पे जितना ज़्यादा cutaneous nerve supply है, so that part has a greater representation in the cortex. Areas of the body which are densely innervated, like face, lips, hands, so they have a greater amount of representation in the cortex in the brain. But remember, the whole body has got a map like this. The entire body is represented. So anywhere from your body, if there is a sensation, be it anything, touch, pain, temperature, it will eventually make its way to its location in the cortex. Once there is a activity in the cortex in the areas, the brain knows where from the stimulus was coming, right? You have a similar type of representation in the motor cortex also. So that is called as the motor homunculus. Areas of the body by which you can make precise movements, right? Say fingers, you can make very precise movements, fine movements. Vocal cords and muscles of the face, so they have got a greater representation in the cortex. Motor aspect, so we'll come to that later on when we talk of motor system. So location is identified when a specific area in the sensory homunculus is activated by a impulse or action potential coming from a receptor. So that is one. Now what about if there are 2 or 3 stimuli? How does the brain know, or how do we understand there is not a single stimulus but 2 or 3 different stimulus? So this property is called as sensory acuity or resolution. See, microscope mein you have learnt about resolution. What does it mean? If there are 2 points, our ability to identify 2 points as 2 distinct points, this is called as our ability of resolution. Similarly here also, if you give 2 stimuli very close, right, the ability of our brain to appreciate 2 closely spaced stimuli or objects as separate is called as resolution or acuity of that particular sensation. So you have practical experiments in the coming month where we will do all of this. You will realize that at each there is this minimum distance below which if you give 2, 3 more multiple stimuli, they will perceive it as 1. Let's say on the back, this minimum distance is around 4 to 6 centimeters. Below this, if you put 2 particular points, they will be appreciated as 1. But the same thing for our lips or the fingertips is in millimeters, 2 to 3 millimeters. So for this to happen, the 2 stimuli to be perceived as distinct, they should impinge upon 2 distinct receptor fields. So that is what makes it identified as 2 different stimuli. And this ability is also called as 2-point discrimination. Right? So 2-point discrimination is better in the areas which are densely innervated, right? And it is understood, and it is lesser, the ability is lesser in areas which are sparsely innervated, like the back, the thighs, and all of these areas. Merkel's disc. So I told this some minutes back also. they have got the smallest receptor field. So Merkel's disc play a very important role in 2-point discrimination. And what was this Merkel disc? Was it a deep receptor or a superficial receptor? Superficial. Once having M, I said is a superficial receptor. So it should be like that. अगर receptor deep में है, this will not help you to understand कि वो 2 point पे था 1 point पे. So it has to be superficial. So Merkel's disc is the receptor which helps us to appreciate this particular ability, 2-point discrimination. Lateral inhibition. One more very interesting property in sensory physiology is whenever there is a stimulus in the center. So I'll come to this, let's understand this first. So this is a receptor, this is the receptive field of the receptor, and center mein there is a stimulus. This brown one is a stimulus that is there, right? This is the actual strength of the stimulus. This is the amount of deformation that is there on the skin. Now what this particular receptor and this neuron is doing. So this is the receptor. So this is conveying it eventually to a second order neuron, then to a third, and eventually from the third it will reach the cortex. But additionally what has happened here is this particular neuron, through some inhibitory interneurons, this has inhibited the neurons on the either side, right? तो बीच में जो न्यूरॉन जो एक इंफॉर्मेशन या स्टिमुलस के बारे में इंफॉर्मेशन लेके जा रहा है, so it is inhibiting the neurons on the sides, right? Sometimes the stimulus is actually very fine. Fine means it is very low intensity stimulus, but even then we are able to appreciate that एक stimulus है उधर, right? So this happens because of this property. जो न्यूरॉन इंफॉर्मेशन लेके जा रहा है, it is inhibiting the surrounding areas. How? By the presence of some inhibitory interneurons. So the receptor number B, as you can see, this has got activated by the stimulus. इसका जो न्यूरॉन है, it is carrying information, but because these are inhibited, the perceived stimulus strength is increased now. The actual stimulus strength पे we do not have a control. Think about your daily life. We cannot control what is happening outside the body, right? So CNS has devised its own mechanism. तो ये एक ऐसा तरीका है कि even without touching the stimulus, we are actually increasing the perception of the stimulus. कैसे? कि साइड में जो एरियाज है that becomes inhibited. तो सेंटर और सराउंडिंग के बीच में जो कॉन्ट्रास्ट है, the contrast increases, right? So this is what helps us to identify. Look at the frequency of action potential in this particular neurons. So here you find a lot of impulses. Surrounding areas may even the firing, normal firing rate is less. So contrast has increased. So this is called as lateral inhibition. Lateral or adjoining areas are inhibited such that the perceived strength of stimulus can be enhanced. Okay. Now let's say again I have put a diagram here where you see that so many receptors are there and receptive fields are also mentioned, right? Deeper ones, they can collect information from a greater area on the skin, right? Superficial receptors are having small receptive fields. So fine touch के लिए या 2-point discrimination के लिए which ones are more important? The smaller ones. Deeper ones, because they get information from a larger area of the skin, they do not participate that much in discrimination, 2-point discrimination or localization. So localization or discrimination के लिए, these are the ones which are more important. Why? Because their receptive fields are smaller. So point to point जो signal fidelity है, precision है, that is better when the receptive fields are small. Now how does intensity of stimulus get coded? So there are two mechanisms. One is frequency coding. So larger the strength of the stimulus, greater the magnitude of the receptor potential, right? And more the number of action potentials that are produced. So this is called as frequency coding, right? We have talked about this during the transduction process. Second one is population coding. What does this mean? as the stimulus strength increases, it tends to spread over a larger area, right? And when it, suppose you think of a prick using a pencil. So if it is a smaller or a low intensity impact, so small area gets deformed on the skin. But if it is a greater intensity, so deformation on the skin is over a larger area. So when a larger area gets involved in this whole process, it either recruits surrounding sensory units, or if it recruits more receptors in that particular same sensory unit, then also number of action potentials will eventually increase. तो दो चीज़ें हो सकती है. First thing is it has spread to a larger area by which आसपास के sensory units have been involved in this whole process. So eventually brain तक जितने action potentials are reaching, they have increased. क्यों? So suppose initially it was going from a one sensory unit, now it has involved 2 or 3 sensory units. Number of impulses have increased. It may also happen that the stimulus is stronger, but not that stronger कि वो आसपास के sensory units को भी involve कर रहा है. It can involve surrounding receptors in the same sensory unit. But even then also, वही sensory units से जितने impulses जा रहे हैं, that will increase. So that is how the brain understands what was the intensity of the stimulus. So this is an example, right? So you see initially there is no response. Gradually, as that deformation increases, the number of action potentials in the nerve increase, and this is where significantly you can see the contrast, this and this. So the intensity has increased, you see the number of action potentials that is there in the nerve. Okay. Now from this, tell me which one among this has a stronger stimulus, A or B? So this is the receptor, these are the afferent neurons, and the small lines that you see, these are nerve traffic, nerve signals. So which one is stronger, A or B? A, right? So you look at the frequency of impulse, that is important. So A is stronger. The duration of stimulus. So duration of stimulus is identified or appreciated by the start and the end point of receptor activity. Here comes the importance of adaptation, right? So adaptation is the process by which if you give a constant strength of current, the number of action potentials will start declining, right? We say that the receptor has become used to, or the receptor has become adapted to the particular stimulus. This is called as adaptation. If this process happens very fast, rapidly adapting receptors, right? So touch is an example of rapidly adapting receptor. Vibration also. Slowly adapting receptors are called as tonic receptors, right? Examples being deep pressure, right, and proprioceptors, right? So where the adaptation process is very minimal. So nociceptors, that is the ones detecting pain, vestibular receptors and muscle spindles show minimal or no adaptation. That means stimulus jitna bhi rahe, the activity is always there. So in both these cases, these are the ones, slowly adapting receptors, which make us feel the stimulus continuously. So this you have to understand. Nociceptors detect pain. Do you think it's important to, for us to be constantly aware of the pain? Is it important or not? It is important, because pain kab hoga? Pain is a protective mechanism of the body. Whenever anything goes wrong deep in the body or from outside, you get some injured, the pain sensation is produced, unpleasant sensation. But this is a warning signal for the body. So this type of receptors, it is for our own good that they should not be adapted. Agar वो adapt हो गया, तो you will stop feeling the pain, and the stimulus could still be there. right? So appendicitis, I'm giving an example. The appendix gets inflamed, it gets distended, you have severe pain in and around the umbilicus and the McBurney's point, severe pain. So if you think of a scenario where pain receptors adapt ho jate, what would have happened? So you would not have been aware that there is something wrong, and the process would have still continued, leading to very serious complications. That is why it is good that they do not adapt. Vestibular receptors, receptors present in the inner ear, utricle, saccule. So these are receptors which help us to at all points of time be aware of the orientation of our body, right? Whether it is erect, whether it is supine. इसमें और mechanisms भी हैं, but the main fulcrum of detection of our body position is vestibular receptors, the ones which detect gravity, as I said yesterday. So it is for our own good that they do not adapt. Muscle spindles. Deep within the muscle there are small structures which are able to detect the length of the muscle. Right? So these are spindles which are very important for maintenance of muscle tone and posture. So जितने भी receptors adapt नहीं हो रहे, it is for our good. Right? Very vital functions are there. So adaptation process का एक graphical representation you can see here. So this is a slow adaptation process. You can see this is the onset of the stimulus. Stimulus is maintained here, and this is where the stimulus is turned off. Generator potential constantly is maintained, and because generator potential is constantly maintained, nerve में जो action potentials है constantly present है, right? So Merkel cells and Ruffini organs are examples of slowly adapting receptors. Rapid adaptation means you see onset पे there is a generator potential and there are some impulses here. उसके बाद even though the stimulus is maintained, there is no generator potential here and no activity here, fine? So this is what means the receptor is become used to or adapted to the stimulus. Again when stimulus off हो रहा है, so there is again a change. So now there is again a generated potential, again there are few impulses. Rapid adapting receptors, rapidly adapting receptors. So जहाँ जहाँ पे on off we should be able to detect very rapidly. The receptors should be rapidly adapting receptors. Meissner's corpuscle, Pacinian corpuscle. So see the beauty, vibration also. So vibration, as I said, is a touch stimulus being given repeatedly. So vibration के लिए दोनों जो receptors हैं, these are Meissner's and Pacinian, rapidly adapting receptors. One more point that this graph also tells. So receptor adaptation is a property or a phenomena that is seen in the receptor as well as in the nerves, sensory system. So both the CNS, circuits in the CNS as well as the receptor, दोनों participate कर रहे हैं in the process of adaptation. So because we are starting with the topic, we will stick to the mechanisms of the receptor. This will gradually come out in due course of time. So rapidly adapting receptor, how does this process happen? This happens, you can see in this diagram, redistribution of the forces. Initially this was a unstimulated receptor normally present. Now you have given some form of a mechanical stimulus here or force here such that there is a deformation in the layers of the Pacinian corpuscle, lamella of the Pacinian corpuscle. तो इससे हुआ ये कि जो ये bare nerve ending था बीच में, so that has become distorted, and remember this nerve ending had stress sensitive cation channels. The channels over activate, right? The depolarization process has started, receptor potential and then eventually action potential. Now if the force is maintained for some time, what happens is the mechanical forces redistribute in the lamella of the Pacinian corpuscle such that on the bare nerve ending, in the core of the receptor, the pressure is or distortion is relieved. So look at this one and the first one, the nerve ending is undistorted here and in the first case. So the moment, so here receptor is still getting activated, but the nerve ending is not giving any response, right? So why? Because the force has become redistributed in all the lamellae. So this is the mechanism of rapidly adapting receptors. Slowly adapting receptors may be the sodium channels and the calcium channels which take part in the depolarization process. They become inactivated gradually, right? And in some cases calcium dependent potassium channels open. So किसी भी neuron में when potassium channels open, what happens? Potassium channels come out. Potassium is more intracellularly, cell के अंदर ज़्यादा है. So when potassium channels open, potassium will come out, and a positive ion is coming out. That means जितना भी ये depolarization या local response बना था, it will start reducing and coming down, right? Positive charge when it is lost, so it will eventually blunt the receptor potential that was formed. At some point it will stop being significant enough. So this is mechanism for slowly adapting receptors. CNS में जो adaptation हो रहा है, that is happening at the level of the brain. Remember we talked about the properties of habituation in synapses. The class on synapses we talked about one of the properties called as habituation. So habituation is something which is happening at the level of the CNS, right? This is one. Then our brain also has mechanisms by which we are able to selectively focus our attention on a certain stimulus, ignoring the background. So selective attention and inattention. This is also a property and a function which is seen by neural circuits around the brain. So both these two processes participate in the process of adaptation happening at the CNS level, right? So with this we come to the close of this particular topic, right? Receptors. So what have we learned here? What is a receptor? It is a transducer, right? Converting all forms of stimuli to action potentials eventually. Electrical response. Initial electrical response is a local potential which gets converted into action potential in the first node of Ranvier, and then once that is formed, it will keep on propagating to eventually to the cortex. Receptors classification is again a very important thing which is again repeatedly asked. You can classify receptors on the basis of source of the stimulus, we can classify on the basis of stimulus energy, and we can classify on the basis of adaptation, right? You will find some more classification in your books. Some people have also classified as superficial and deep, right? So ये बहुत सारी चीजें हो सकती है, but the three main ones are source of the stimulus, energy of the stimulus, and adaptation, right? Okay. And in bits and pieces, receptor adaptation is asked sometimes as a short note in your exams. You might be asked to differentiate between a rapidly adapting and a slowly adapting receptors. ये पूछा जाता है एग्जाम में, right? Lateral inhibition is a very common question, and you will see this concept so many times it comes on. In the coming lectures, you will find the importance of lateral inhibition. This is also a common question. And this one, Muller's doctrine of specific nerve energies. So for short, these are asked, right? So you have any queries or doubts on this topic? All right then, thank you.